Hey guys, so let's keep going and now let's talk about cholesterol metabolism. I would say this is not a big deal for your MCAT, like at all, as you can see. I mean, quite literally, I haven't seen this like at all. But low yield stuff still can pop up on test day. So let's go over it. So cholesterol metabolism happens in the liver and it's called de-Navarro synthesis. And it's dietary, okay? So you can have dietary cholesterol metabolism and then you can have de-Navarro synthesis metabolism. These two are separate from each other. So this is probably the most important thing of this chapter is you must know that the rate limiting step of de-Navarro synthesis is going to be HMG-CoA reductase. Okay? And you also need to know insulin promotes synthesis, cholesterol, and stains inhibit it. So there's a negative feedback loop of cholesterol synthesis. So if you have a lot of cholesterol, you're not going to do it. Very, very simple feedback loop, right? So transport. LCAT adds fatty acids to cholesterol, and then that's how you get cholesterol esters, and then CEPT transfers esters to other lipoproteins. And that's it. Definitely, I think the only thing I have seen on my full length so far is probably the rate limiting one. So next, let's go over fatty acids and triglyceride. So let's go over nomenclature first. So it's based on the chain length and the double bond position. Okay. So one thing, right? This is pretty cool, I guess. So when you hear omega-3 fatty acids, omega-3 is going to tell you the double bond is on carbon 3. Okay. So that's just one of the ways that we can name the double bond position. And then you can have different omega type of fat, and so on and so on, all right? So synthesis. So fatty acid synthesis occurs mainly in the liver and sometimes in your adipose tissue. It requires acetyl-CoA and NADPH. Steps. Citrate shuttle moves acetyl-CoA from mitochondria to the cytosol. Acetyl-CoA carboxylase forms malonyl-CoA, and again, you should always know your rate limiting steps. Fatty acid synthesis adds 2 carbon units using NADPH. Okay, and these are the really the most important steps you need to know. And I would actually know all of these by heart. This is relatively important, just in general. And triglyceride formation. So triglycerides are formed in the liver, and then they're packaged into VLDLs for transport. And again, we know VLDLs are a type of lipotransport protein. Okay, so let's keep going. Beta oxidation, very important. So you need to know it occurs in the mitochondria, and then your creatine shuttle brings long fatty acids into the mitochondria. Each cycle removes 2 carbons and makes those 2 carbons acetyl-CoA. So you take 2 carbons as acetyl-CoA, and then you produce NADH plus FADH2. And let's go over ketone bodies really quickly. So ketoneogenesis. So you really use ketone bodies in forms of like starvation, right? So if you are fasting or something and you don't have glucose, your glucose would then your brain needs some sort of energy, right? And that's when we start forming ketone bodies. As we see here, right? It occurs in liver mitochondria during fasting or prolonged starvation, and it occurs because you don't have glucose. So you can turn acetyl-CoA into ketone bodies. These are 2 types of ketone bodies, and then another one is going to be acetone, and we all know how acetone looks, okay? So these are your 3 ketone bodies, and I would know that by heart. And ketolysis, and again, lysis means breakdown. So when we're breaking down ketone bodies, it happens in the tissues, especially, again, like I said before, in the brain, right? If your brain doesn't have glucose, your brain will use the ketone bodies. It converts ketones into acetyl-CoA. And liver cannot use ketones. Okay, liver cannot use ketones.